Hi, and welcome to Chapter 12. In this chapter, we're looking at Mendelian genetics. Before Gregor Mendel's work became more well known, there were many misconceptions about heredity. One of the most commonly held misconceptions was blending inheritance, meaning that people thought the offspring of individuals were the average between both parents. So people thought that original parental traits were blended in the offspring. And now we know that's not the case because we inherit characteristics as discrete units. And we're going to see that is the main topic of Chapter 12. Something else that we thought was not true, and then nowadays we think may be actually true, is that changes happening to parents, so changes happening to someone who is an adult, could be passed on to their offspring. And this is going beyond genetics into the field of epigenetics. We're going to learn more about this in the article that we're going to be reading together in the class, as well as in future chapters. So Gregor Mendel is considered often to be the father of genetics, and he studied pea plants as his model system. In fact, in around 1865, he presented these results of experiments that he conducted on over 30,000 pea plants to the Natural History Society, and he showed that traits are transmitted discretely from parents to offspring, and each trait is transmitted independently of other traits, and there were clear dominant and recessive patterns that we'll talk about in this chapter. So why did Gregor Mendel choose peas? There are many reasons, including that these pea plants have a lot of different dichotomous traits. For example, in the characteristic of flower color, the pea plants could either have purple flowers or white flowers. For seed shape, they were either smooth seeds or wrinkled seeds. For seed color, some of them were yellow, others were green. So these were dichotomous traits where you see one or the other trait for each of these characteristics. Many of these traits were true breeding, and that means, for example, for a purple flower, they would contain only the genes for purple color. So that could be purple gene, another purple gene. And then, for example, a white true breeding flower could be, usually would use a lowercase p. So they would have lowercase p for the white flower and another lowercase p for the white flower gene. Pea plants are also monoecious. Sometimes this is known as being hermaphroditic or hermaphrodite in terms of an organism, where male and female organs are in a single organism. This makes it really easy to manipulate the matings in these pea plants. Each pea plant is a new individual, so you can evaluate thousands of offspring. And they have short generation times. The garden pea grows to maturity, for example, in one season. Also, these are quite affordable, and they were readily available back then too. We're going to be following Mendel's notation system throughout the chapter, and also in the practice problems we'll be completing in the lab. So the P generation is the parental generation, and we're going to follow the standard that the parents will always be true breeding. This means that they will always be homozygous for some kind of characteristic. And let me give an example of purple flowers, purple flower parents in the parental generation. They will have two of the same alleles, so two of the same genes, both of them encode purple flowers. And then a parent plant with white flowers will have lowercase p, and these are also true breeding. They have two of the same alleles, both encoding the gene for white flower color. If two parents cross and mate and you produce some offspring, that offspring is in the first filial generation, where filial refers to offspring or daughter or son. So that would be the F1 generation. And here it would be, for example, a big P, small p, child. If you take two F1s and you mate those two, so let's pretend I have two big P, small p, another big P, small p, and I mate those, Then you get the next generation. That next generation would be the F2 generation, the second filial generation, and so forth. If you keep crossing them, you would have successive generations. If you ever see the term self-cross, this is when you cross a plant with itself, and that's possible because these are monoecious organisms. So for example, if I took my F1 plant, which has a big P allele and a small p, I could cross it with itself, and that would be a self-cross. A test cross is when you have some kind of organism, some kind of pea plant, for example, and you cross it with something that's known as a homozygous recessive plant. So in the case of flower color, this would be with a lowercase p and a lowercase p. This would be the white flower. That would be the tester. That's the homozygous recessive organism. And then true breeding, I mentioned earlier. True breeding, we're going to see those in the parental generation. When parents having the same traits, always produce offspring with the same traits. So if I had purple flower mate with another purple flower, all of their offspring will also have only purple flowers. Here's an example of a true breeding parent where both of them are true breeding for purple flower color. So parent 1 has big P. They're purple. The second parent is also big P. And they can only generate purple flower offspring. So there will be no variation in the genes for these offspring. They're all going to be true breeding. Large P the whole time. But then Mendel tried something different. He crossed parent plants that differed in a trait to produce hybrid strains. So let me show you an example here. We have parent 1. That's a true breeding purple plant. So let's say that's big P in terms of its genes or genotype. Then we have our true breeding white plant, which is small p. And if you cross these, you would get the F1, the first filial generation. Their offspring would be big P, small p, because remember in meiosis, you get one gamete from each parent. The other child would also be big P, small p, or this could be a self-cross. And this is called a heterozygous genotype, because you have one of each allele, a big P and a small p. In contrast, the true breeding parents, these were homozygous. So homozygous is when you have two identical alleles. This one on the left is also homozygous. Another term for heterozygous, sometimes you'll hear these are hybrid individuals, again, because you have a large or a big P, one allele, and the second allele is different. And then when they crossed two heterozygous individuals, or you could say two hybrid individuals, you get a different ratio of purple and white flowers in the F2 generation and the second filial generation. This cross, the big P small p crossed with another big P small p, is called a monohybrid cross. Hybrid because you're looking at a heterozygous individual, and mono because you're only looking at one characteristic. You're looking at flower color here. We're not looking at how tall the pea plants are or what kind of seeds they have. We're just looking at flower color. So mono referring to one characteristic, and hybrid because the cross is between heterozygous individuals. This is the same experiment we just saw, but using a different kind of picture. We had purple plants, and these were true breeding, purple flowers. This one was the white flower true breeding plant, small p. They crossed these two, and they got an F1 generation that only had purple flowers. And these we saw that they were heterozygous, they were hybrids. Then they crossed these, so this was through self-fertilization. So this is a self-cross, big P, small p, crossed with another big P, small p. Remember, this is called a monohybrid cross. And what Gregor Mendel found was he always got a 3-to-1 ratio of the purple flowers versus the white flowers. So how do you get 3-to-1? 705 plus 224. What is that? That's 929. So 705 over 929 versus 224 over 929. That would give you a 3-to-1 ratio. So 75% of the offspring for the F2 generation were purple flowers or had purple flowers, and 25% had white flowers. This is always going to be true for a phenotypic ratio, phenotypic ratio. Phenotypic ratio meaning the colors you see, what you actually see, what's observed. So this 3-to-1 phenotypic ratio will always apply for our standard monohybrid crosses. So what Gregor Mendel concluded from these crosses was that blending inheritance was not happening, was not the case. Instead, inheritance was particulate. And we know today that we inherit genes, discrete genes from each of our parents. As he saw, some versions of a trait masked other versions. For example, those purple flowers were dominant to the recessive white flowers that we saw earlier. The recessive trait can sometimes disappear in some generation to reappear later on, as we saw in the heterozygous F1 generation, but then it reappeared later in the F2 generation. And remember from standard monohybrid crosses, where you have something like a big P, small p crossed with another big P, small p, you get a 3-to-1 dominant to recessive phenotypic ratio, what you see. So these ratios suggested that these hereditary particles, these genes or alleles, segregate into gametes and then they come back together during fertilization. And we saw this during meiosis. Remember, our cell starts off as diploid. Let's say this is our diploid organism. Remember, at the end of meiosis, you have haploid gametes. This could be one of your gametes, big P, and the other one could be small p, and that would be haploid. Here are a few more terms that are important for Chapter 12, including trait. So when we look at characteristics like flower color, we saw traits such as purple flowers versus white flowers. For eye color, we have different traits, and you can have more than two. It doesn't have to be dichotomous. So we could have, for example, brown eyes, hazel eyes, blue eyes, etc. Phenotype is what we see, what you can observe. For example, purple flower or blue eyes, if we're talking about these characteristics. Alleles are the versions of genes that we inherit from our parents. For example, some alleles are dominant, like in the purple flower. We might inherit big P from one of the plants, and another plant might inherit the small p from the other parent, and that would be the recessive allele. Your genotype is the combination of alleles or genes that you've inherited from your parent. For example, if I write big P, small p, this would be the genotype for a purple flower. But the phenotype, if I'm looking at phenotype, I would say that's purple. So phenotype would be purple, the genotype would be big P, small p. Homozygous individuals, as I mentioned earlier, have the same alleles for both of the traits, what they inherited from each parent. So big P is a homozygous dominant individual. Small p is a homozygous recessive individual, and these are also are always true breeding. Heterozygous individuals, in contrast, have two different alleles, like big P, small p. Zygotes, remember a zygote is the first cell that's formed after fertilization. So this is a diploid cell. All right, lots of vocabulary here. Another set of terms. We have genes that we've seen before. We inherit genes as discrete particles from our parents. Let's say I have a chromosome here. Here's a sister chromatid attached at the centromere. And let's say right here is the gene for flower color in the pea plant. So the gene would be located here, and the locus, the location of the gene, we call the locus. For this gene for flower color, remember we could either have big P for purple flowers, and that would be one version of the gene, or you could have small p for white flower color as the allele. Reciprocal crosses occur when you repeat a cross, but you switch parents. So let's say I have a purple plant, and that's true breeding, and that's the mom, and I have a white flower, and that's the dad, and I get purple offspring, and that could be either male or female. A reciprocal cross would be I do the same thing, except this time my purple flowers are from dad and my white flowers are from mom, and I would still get my F1. If there's no difference when you perform a reciprocal cross, this usually tells you that the allele or the gene is not located on the X or the Y chromosome. It's usually instead located on chromosomes 1 through 22, one of those chromosomes, and we call these autosomal genes, if they are not related to sex, X or Y. If they are on the X or Y chromosome, we're going to call those sex-linked genes. So again, if a gene is on chromosomes 1 through 22 in the human, we call those autosomal genes. If the gene will be located on the X or Y chromosome, we're going to call those sex-linked genes, and we're going to see that later on. Two more terms that are important include the wild-type allele. Wild-type we often write just WT. The wild-type allele is the most common allele that we find. Sometimes we also call it the normal allele. Whereas the mutant allele is rare, and it arises by mutation. So just because something is a mutant allele does not necessarily mean that it's recessive. Either of these could be dominant or recessive. So let me try to picture what alleles look like on the chromosome. So remember, we often see chromosomes as an X-shape structure. Remember, on one side, this is one sister chromatid, and this is the identical sister chromatid on the other side. Oh, that's a really ugly picture. But let me look at just one of these chromosomes without its identical copy, and I have that over here. So that's one of those chromosomes. And here it looks like right here, I'm saying that's where the location of the flower color gene is. And maybe from the other parent, you have the same or the homologous chromosome. And again, the gene would be located at the same place, the same locus, except we have a different version. We have an allele for white flowers in this case. What's the difference between the two versions of this gene for flower color? It's usually in the sequence of nucleotides that we have in the DNA. So in terms of flower color, this sequence of nucleotides allows the formation of an enzyme that synthesizes purple pigment, whereas if you have the little p allele, then that sequence of nucleotides does not allow that enzyme to be produced. So as long as you have one big P allele, then you make enough enzyme to synthesize the purple flower color. All right, to finish up the first part of the video, I just wanted to go over those genotypes with you again. So this is the parental generation. We had true breeding parents, both homozygous. This is homozygous dominant, homozygous recessive. Remember, the F1 generation from these true breeding parents was a heterozygous or hybrid individual. And if you self-cross the hybrid individual using a monohybrid cross, remember that we get a 3-to-1 phenotypic ratio. What you see, remember this is what you see. And if I look at the genotypes that I actually get, how do I get the genotypes? The offspring, the F2 generation, will have or can get a big P from one parent and the big P from the other parent, and that's how you get the first purple flower. Or they could get a big P from parent 1, but a small p from parent 2. Or they can get a small p from the first parent, but a big P from the second parent. Or finally, they can inherit a small p or small p from both parents. So the phenotypic ratio from a monohybrid cross is 3 to 1, but the genotypic ratio is 1 to 2 to 1. That would be the genotypic ratio. You get 1 homozygous dominant, 2 heterozygous individuals, and 1 homozygous recessive. And again, that's the genotypic ratio. That's the phenotypic ratio. And I would like you guys to memorize these ratios for the monohybrid cross. That takes us to the end of the first video. In the second video, we're going to talk more about test crosses and when we can use the multiplication and addition rules and probability.